So we discussed that the extracellular matrix consists of protein fibers, such as collagen, okay, and ground substance. And this ground substance may contain glycosaminoglycans, proteoglycans, and stuff like that. So protein fibers, we're going to do these first, and what protein fibers the ECM may contain and how they're formed and what forms them. So number one would obviously be the most abundant, which is the collagen, which is the primary protein of the connective tissue. All right, so collagen is the first fibrous protein that we're going to study in that is present in the ECM. Let me just go over here into this neat note table. So collagen is a fibrous protein, and it is acidophilic, so it would bind with the eosin dye and appear pink on a microscopic slide. Collagen can be divided into three different characteristics according to its three different characteristics, either fibrillar, network forming, or binding collagen. There's three types of fibrillar collagens. Type 1, which is mostly present in your bone. So if someone says type 1 collagen, you just refer to bone, but it's also present in tendons and skin. The associated disease is osteogenesis imperfecta. We're going to study how that works in a bit, but this is also called a brittle bone disease. Brittle bone disease, osteogenesis imperfecta. Genesis basically means the generation of something, the production of something, and imperfecta means imperfect. So imperfect generation of bones, or also the brittle bone disease, in which there's bone deformity, pain, and fractures, especially fractures and brittle bone disease. The bones are brittle and they're easily fractured. Type 2 collagen is present in cartilage and inside the vitreous humor of the eye. So let me just show you where the vitreous humor is. So this would be your eyeball, right? And the liquid inside the eyeball, this big liquid right here, would be your vitreous humor. It's a pinkish liquid that contains type 2 collagen, all right? And it's also present in the cartilage. Type 3 collagen is present in your blood vessels, inside your granulation tissue, which is a tissue that is formed at the sites of chronic inflammation, and it gets supplied by blood vessels, and is also found in the blood vessels. So any problem with the type 3 collagen would lead to obviously a disease in the blood vessels. And here you can see it's got pathology in the aortic intestinal rupture, because type 3 collagen is mutated in this disease, and it causes the aortic intestinal rupture. It is also characterized Ehlers-Danlos type 4 disease. It's also characterized with translucent skin. Over here, you can see that the fibrillar type 1 collagen is present in bone, and when mutated, it causes this disease, which is related to the bone as well. Moving on to the network forming collagen, which is going to be the type 4 collagen. It is present in the basement membranes. The basement membrane, remember I told you one of the major sites of basement membrane and its function is where the exchange of materials is taking place, for example, the kidneys. And here we have a disease that is related to kidneys. You might study this in biochemistry, and we'll make sure we cover this if we do a biochemistry series as well. And epidermolysis bullosa inside the beta integrin mutation, we did this in the last chapter. So any problems with the type 4 collagen, which is the network forming collagen, would lead to a disease in the kidneys or a disease in the skin, basically a disease in the basement membrane. Now the binding collagen, this binds a type 4 collagen and type 3 collagen and type 4 collagen especially, so it would lead to a disease in the basement membrane again. And we study the type 7 collagen is present in the basement membrane in the first chapter, the epithelium. All right, so next up is the synthesis of collagen. Even though the synthesis of collagen is concerned mostly with biochemistry and cell biology, but we have to do this in histology as well. And the reason for that is because there's pathology associated with it. And to better understand that pathology, we have to do the synthesis, so we can relate to what's going wrong in the synthesis, which is causing certain diseases. So as collagen is a fibrillar protein, then the synthesis of collagen would also be just like any other protein, and that is through mRNA and then mRNA into polypeptides. So as we learned that collagen is synthesized in the fibroblasts, so this would be your fibroblast right here, and then it would also have a nucleus, and inside the nucleus there's a thin strands called chromatin that contain the DNA, and then this DNA is transcribed into mRNA, and then mRNA, and then mRNA is translated into a polypeptide, all right? And this happens in the ribosome, and then this polypeptide, what it does is that it enters the ER. So let me just write ER right here so that we know that it's entering now the endoplasmic reticulum. This polypeptide of collagen is a triple repeat or a repeat tripeptide of glycine. So every third amino acid would be glycine, all right? Every third amino acid would be glycine. And it would this mRNA would be translated into a pre-pro-alpha chain. So this chain would be called a pre-pro alpha chain, right? And it contains a hydrophobic terminus right here, which allows it to enter the ER. So this hydrophobic terminus, when it is cleaved inside the ER, it is now referred to as the pro-alpha chain. This right here is cleaved. And any problems with the cleaving would lead to osteogenesis imperfecta. We've studied it up here. Let me just show you. So this right here, osteogenesis imperfecta, which is the brittle bone disease, which affects the type 1 collagen. So moving on, we have the hydroxylation of this chain. So what happens is that there's enzymes called proline and lysine hydroxylases that attach a hydroxyl group to the proline and lysine residues on this chain. And this hydroxylation is vitamin C dependent. Very important to know. Let me just highlight this real quick. Vitamin C dependent, especially lysine hydroxylase, because there's pathology associated with it. So any problem with this hydroxylation would lead to pathology, especially concerning collagen. So for example, if we have a deficiency of vitamin C, then that will lead to a disease called scurvy. Might have studied this earlier. This is a disease that is characterized by bleeding of gums and loose teeth, and also a little bit of anemia. And then lysine hydroxylation problem would lead to a disease called Ehlers-Danlos syndrome. We have studied this earlier. This is characterized by hypermobile joints and pullable skin and a very loose connective tissue. And of course the arterial rupture in varicose veins that we discussed earlier. All right, so what happens next is that these hydroxylated chains are then N-glycosylated on asparagine residues. If there's N-glycosylation, you will study what that is in biochemistry, but there's asparagine residues on this polypeptide chain on which in the endoplasmic reticulum there's N-glycosylation, and then there's a combination of hydroxylated and glycosylated polypeptide. And these polypeptides are then arranged in a triple helical structure. So this would be a triple helix still in the ER, and then after it is formed into this triple helix, it will then be secreted into the cytoplasm. So now finally, after we have the triple helix, let me just write that down, triple helix, then it will be secreted into the cytoplasm, and here it enters the cytoplasm. All right. In the cytoplasm, it is then again cleaved. This would again be cleaved, and then a very major step that happens is that there's oxidization on lysine and hydroxylysine residues. Which allows these triple helixes that are now cleaved to be arranged into fibrils. So these will be arranged into fibrils, and this oxidization is dependent on copper, by the way. So any problems with copper would lead to the oxidization not happening correctly, and then this would lead to a problem in forming the collagen fibrils, and this would of course lead to pathology. So a disease associated with copper deficiency and collagen is called Menkes disease. And this disease is characterized by arterial tortuosity syndrome, which is basically the lengthening and twisting of arteries. And why is that happening? Because in the arterial connective tissue we have collagen, and collagen is not being synthesized properly. Why is it not synthesized properly? Is because we don't have copper. There's copper deficiency, which results in a problem in the oxidization of lysine hydroxylation, which results in a problem in the collagen synthesis, which leads to the arterial tortuosity syndrome, which is basically the lengthening and twisting of arteries. So that is the major symptom or sign for the Menkes disease. Another major sign for the Menkes disease would be depigmented hair. Depigmented hair because the pigmentation of hair requires melanin production, and for producing melanin we need copper. So copper is required for melanin production, and when copper is absent, it is deficient, then there's also depigmentation of hair together with the arterial tortuosity syndrome, which is the lengthening and twisting of arteries. And this is the fibril form, and then these fibrils are then accumulated. So these would be accumulated. Let me just something like this, and these would be your mature collagen that would be then secreted out of the cell. And that's it for the synthesis of collagen. Congratulations. So let me go over what we studied real quick. We studied that inside the extracellular matrix there's protein fibers, and there's also the ground substance. The protein fibers comprise mainly of collagen. Collagen can be divided into three characteristics: the fibrillar collagen, network forming, and binding collagen. Fibrillar is the most important. It has three collagen types: type 1 collagen, which is associated with the bones and present in bones; type 2 collagen, which is associated with cartilage and intratumor; and type 3 collagen with blood vessels and granulation tissue; and then performin, which is present in the basement membrane, the type 4 collagen; and then binding collagen, that is type 7 collagen. And then we studied the synthesis of collagen. Is that collagen is synthesized just like any other proteins. It contains it is a repeat tripeptide of glycine residues, and then a pre-pro-alpha chain is formed, and then the pre-pro-alpha chain is cleaved on its hydrophobic terminus right here, and then the pre-pro-alpha chain is then hydroxylated on proline and lysine residues, hydroxylated. And any problem in the and this hydroxylation is dependent on vitamin C. So any problems with the vitamin C or hydroxylation enzymes would lead to pathology like scurvy and Ehlers-Danlos syndrome. And then after hydroxylation, it is glycosylated. This is N-glycosylation. And then it is arranged into a triple helix form, and these triple helices are then excreted into the cytoplasm, where they are cleaved. And inside, after cleaving, inside the cytoplasm, there's oxidation of the lysine and hydroxylysine residues by enzymes called lysyl oxidases, and this oxidation is dependent on copper. So any problems with the copper or the enzyme not working would lead to pathology concerning collagen. And one of the pathologies is the Menkes disease, one of the most common pathologies. You might see this in the USMLEs. And then those fibrils are then aggregated to form the mature collagen. So we finished the major constituent of the extracellular matrix, that is collagen, and next up is reticulin. Reticulin we've studied before. It is type 3 collagen. The reason why we're doing it separately is because it is stained black with silver salts. Unlike collagen, which was stained pink, remember, it is stained black with silver salts. And it is made of type 3 collagen. We've studied this before, and it is present in the reticular lamina of basement membrane. So any problem with the type 3 collagen would lead to problems in the basement membranes. And reticular connective tissue, which we will study in a bit. And the final fibrous protein that is found in the extracellular matrix is the elastin. Elastin aggregates and joins up to form the elastic fibers that are present in the connective tissue. Especially those connective tissues or those areas which have a need for elasticity in their nature. So, for example, inside the walls of the arteries, you would find elastin and elastic fibers being present inside the earlobes, inside the lung stroma as well, because those organs are elastic in nature. Now, the difference between collagen and elastin on a histology slide would be something like the width of the fiber. So collagen would generally be thicker than elastin, and elastin will be more densely stained than collagen, but they will have, so for example, if collagen is stained light pink, then elastin will be stained in a darker pink. So let's assume that this right here would be collagen, and then this would be elastin, right? And elastin is a protein or fiber that is made up of a protein called fibrillin, which joins up to form microfibrils, and then these join up to form the elastin or elastin fibers. Now, fibrillin is important to know, and the reason for that is because of the Marfan syndrome. Now, because fibrillin is a protein, it is coded from the DNA, and on chromosome 15, although there's a little bit of medical genetics involved, on chromosome 15 we have a sequence of DNA that codes for fibrillin. Any mutation in that would lead to fibrillin not being produced correctly, and this would lead to pathology in all the connective tissues that have elastin associated with them. So, for example, the arteries, especially the large arteries like the aorta, would get pathology in there, and that's a very major symptom of the Marfan syndrome. Is that there's aortic aneurysms that are found in patients having this disease. The enzymes that degrade elastin would be elastases, just like the degradation of collagen would be through collagenases. And that's pretty much it for the protein fibers. And next up we have the ground substance.